Bello. Today, our group investigated a very important scientific question. Almost as important as how many bananas can a minion eat in 1 minute. Our experiment tested which sugar helps yeast ferment faster, glucose or sucrose. Fermentation is a process where yeast break down sugar without oxygen and release carbon dioxide gas, or CO2, kind of like tiny yeast burps. For our hypothesis, we predicted that glucose would ferment faster than sucrose, because glucose is a simple sugar that yeast can use right away. Sucrose, on the other hand, is a disaccharide, so it needs to be broken down first before yeast can use it. So, in banana translation, we expected glucose to make the yeast go be-do much faster. We prepared for test tubes with yeast mixtures. Tube 1 had live yeast and distilled water, and this acted as a negative control, because there was no sugar for the yeast to ferment. Tube 2 had live yeast with glucose solution, our first test sugar. Tube 3 had live yeast with sucrose solution, our second test sugar. And Tube 4 had glucose added, but with boiled, also known as dead yeast, which served as another negative control, because the yeast enzymes were no longer active. All tubes started with 10 milliliters of yeast, and we observed them over time to compare how much CO2 each one produced. If fermentation occurred, we would see bubbles and foam forming at the top of the mixture. We recorded the gas production in each test tube over time, checking and measuring the height of the foam, or CO2 gas layer, at regular intervals. As the yeast fermented, the bubbles increased, and we documented the changes with both observations and measurements. Tube 2 and Tube 3 showed visible bubble formation, while Tube 1 and Tube 4 mostly stayed flat and unbubbly, like soda that's been left open for 3 days. We collected our data, organized it into a table, and then graphed the results to clearly show the differences in CO2 production. Our results showed that glucose fermented the fastest. Tube 2, with glucose, produced the greatest height of CO2 bubbles over time. Tube 3, with sucrose, did show fermentation, but at a slower rate and with a lower total CO2 height. Tube 1, the water control, produced virtually no CO2 since the yeast didn't have a sugar source. Tube 4, with boiled yeast and glucose, also showed no fermentation because the yeast cells were no longer alive to perform the process. So, our results supported our hypothesis. Glucose led to faster and greater fermentation than sucrose. These results make biological sense. Yeast can use glucose immediately for glycolysis, the first step in fermentation, so CO2 is produced more quickly. Sucrose takes longer because yeast must first break it down into glucose and fructose before fermentation can begin. This delay reduces the rate of CO2 production. Now, quick fun comparison. Yeast do alcoholic fermentation, which produces ethanol and CO2. Humans, when we do anaerobic respiration, like during intense exercise, perform lactic acid fermentation, which produces lactic acid instead of alcohol. That's why your muscles burn after sprinting, but yeast party and make bread rise instead. This video was created by our group, Melvin, Lemery, Sandy, Ruchan, and Tristan. Thank you for watching, and remember, science is almost as fun as bananas. Be-do, end of presentation, and happy Halloween!